So now it's time to talk about DNA research. Um, DNA research goes back to actually the 1800s. Um, Friedrich Miescher, German scientist, actually is credited with discovering DNA, and he basically called it nuclein inside the nucleus of cells. And in the late 1800s, scientists kind of figured that it was the chromosomes in sperms and eggs that actually carried the genetic information that was passed to the offspring. The only thing is, though, chromosomes actually contain both DNA and protein, and the question was which one was hereditary, and the answer was not self-evident. So by the late 1940s, it was shown that it was DNA, but believe it or not, at the turn of the century, most biologists believed protein was responsible for heredity because protein was a lot more complicated than DNA and was believed to be capable of carrying hereditary information. So we start in with a very important piece of research that was being done in the 1920s. Uh, the phenomenon that was discovered through this research is bacterial transformation. In 1928, British scientist Fred Griffith published a paper about some peculiar findings in his studies of bacterial pneumonia. Fred was looking for a vaccine to the deadly disease pneumonia, and he was injecting mice with different strains of pneumonia to see if he could develop a vaccine. So Griffith basically found two different strains of pneumonia. One of them extremely difficult to work with because it caused pneumonia and it killed mice. It was the virulent S strain, it's called. And he also discovered another strain that did not kill mice when injected. It was called the non-virulent or R strain. He was pretty excited about that R strain initially, but it did not actually confer vaccination when it was used in an injection. What Fred discovered, and this graphic here kind of summarizes, if you injected dead virulent and living non-virulent, by the way, this is not unusual. Vaccinologists often kill bacteria and then attempt to see if the dead bacteria, when injected into a living thing, will induce vaccination. What that means is if they kill some bacteria, inject them into you, you then would become immune to the bacteria that you were injected with, even though they're dead. In many cases, those dead bacteria will cause your immune system to respond, and you will become immune to that strain. So should you ever run into the living bacteria, you don't get the disease. So what Fred found is if he injected dead virulent and living non-virulent, some of the mice would die of virulent pneumonia. Now that was very confusing. So the bacteria that were virulent were dead, so they can't wake up and kill the mice. He figured somehow these dead virulents transformed the living non-virulents into lethal strain, and that's what killed mice. So ultimately now, understand the rough strain is non-virulent. When you inject that into mice, they don't die. The smooth strain is lethal. When you inject that into mice, notice the lovely graphic of the dead mouse. If you heat kill the smooth strain, unfortunately, it doesn't give it won't cause vaccination, but the mice don't die. If you inject heat-killed smooth bacteria into mice, they do not die. They actually live. The weird part is if you inject living rough and heat-killed smooth, a number of the mice die of pneumonia. Now that's weird. So that was very unexpected. Fred was not banking on that happen. But his conclusion was that some genetic material of the dead virulent bacteria transformed the non-virulent bacteria into virulent ones. Fred did not get to determine what the transformation factor was because he was killed in a bombing raid on London by German planes. So Fred did not get to find out what it was. But after Fred had died in the 1940s, around 1944 actually, Oswald Avery, Colin McLeod, and Macklin McCarty, working together, showed that the only transforming factor in the dead bacteria was DNA. What they did was they separated dead smooth bacteria into DNA, protein, carbohydrate, or lipid fractions. And they then would test the DNA of the dead bacteria separately from the protein, separately from the carbohydrates, separately from the lipid. The only chemical they could get to transform the rough strain of the bacteria into smooth was DNA. But still, there were a lot of skeptical biologists because so many people at the turn of the century believed that protein was the hereditary chemical. So there was a later experiment done by Alfred Hershey and Martha Chase, which went on to show that it actually is DNA, at least or nucleic acid in viruses, that transmits hereditary information. And by the early 1950s, many biologists had switched over into accepting the idea that DNA was the hereditary chemical. Now that brings us to Watson and Crick. So in the early 1950s, Rosalind Franklin, doing intense research on DNA with X-rays, was producing crystallographs. Rosalind Franklin had crystallographs that looked like DNA was some kind of a stranded molecule that was twisted around, but she couldn't figure out quickly what the structure was. Unfortunately for Rosalind, a common lab researcher, Maurice Wilkins, passed along some of her actual work to James Watson and Francis Crick. And they, in a matter of maybe 2 weeks, determined the structure of DNA and published a quick paper in 1953 that basically laid out the structure of DNA. Their paper basically presented the idea that DNA was two strands. It was basically like a ladder, and it was twisted into something they call a helix. The ladder sides were sugar and phosphate molecules, and the rungs were nitrogen bases. And so basically, another way to look at the DNA was that DNA was two strands of nucleotides. And each nucleotide is made of a sugar, a phosphate, and a nitrogen base. Turns out that the nitrogen bases that Watson and Crick were talking about came in two types: purines, which are adenine and guanine, and pyrimidines, which are thymine and cytosine. The purines are double ring structures, and they're different than the pyrimidines, which only have a single ring structure. We'll see those. This is a purine nitrogen base here, and this is a pyrimidine nitrogen base here. And the way DNA pairs up, it's always purine with pyrimidine. Purines never bond with purines across the DNA, and pyrimidines never bond with pyrimidines. Next, adenine, which is a purine, always bonds with thymine, which is a pyrimidine, and there's only 2 hydrogen bonds between them. Cytosine bonds with guanine, and it has 3 hydrogen bonds between them. The 2 strands were proposed to be anti-parallel to each other, and the 5' phosphate groups of the 2 strands stick out at opposite ends. I'll explain what 3' and 5' are in just a second. The whole idea was that about every 10 nucleotides you would get a complete revolution of the double helix. It turns out that one side of the DNA is what we refer to as the template strand. The template strand, the antisense strand, is actually coded into RNA. It's called antisense. You'll see why later. It's the way DNA is copied. The coding strand is the opposite side, but it's actually not transcribed. But it actually has the nitrogen base letters that actually end up determining the protein amino acid sequence in the protein. But we'll talk more about that in a few minutes. At any rate, the DNA molecule is shaped the way you see in this picture here. Let's see if I can't get this selected. There. I need to scroll here. So let's see. Yeah, here we go. So The way the molecule is built, and let's get a little pen here going again. This is a nucleotide right here. That's called an adenine nucleotide. This right here is another nucleotide right there. And that has 3 hydrogen bonds, and this is a pyrimidine, so this is cytosine nucleotide. This is adenine nucleotide. Notice there's only 2 hydrogen bonds there between the adenine and the thymine. This one down here is a thymine nucleotide bonded to an adenine nucleotide, so the entire nucleotide is there. Okay, and then there's one more nucleotide down here, and this is guanine right there. And then that's a string of nucleotides, and it's the sugar, sugar, phosphate, sugar, phosphate, sugar, phosphate, sugar, phosphate, makes up what we call the backbone. You can see this is the side of the ladder, and these are the rungs of the ladder going across here. Now I've made a complete mess out of the picture, so let's get this back. Okay, now it turns out that we number the carbons of the sugars 1, 2, 3, 4, 5. So this is a carbon here. This oxygen. So wait, let me go over this. This is a carbon here. Carbon, carbon. So carbon number 1, carbon number 2, carbon number 3, carbon number 4, and carbon number 5. The 3' and 5' designation of DNA is referring to the carbons. So they give these primes because the carbons and stuff, nitrogens over here are numbered 1, 2, 3, 4, 5, whatever. So the carbons that are most important here are the number 3 carbon of the sugar and the number 5 carbon of the sugar. You'll notice that all the phosphates here are facing that way, and the phosphates over here are facing this way. And so we call this the 3' end here. There's no phosphate sticking out here, and we call this the 5' end down here because there's a phosphate sticking out here. On the opposite side, they're anti-parallel, and they face the other way. So this is the 3' end with no phosphate, and this is the 5' end with a phosphate sticking out. And this shows the molecule is flat, but in reality, it's actually not flat. This is a better artistic rendition of the molecule, but obviously there's way too much information there to really make too much specific sense of what's in there. This captures a much better essence of the molecule, but this in actuality is more what it actually looks like. And here you can see the adenine. This is adenine nucleotide bonding to thymine nucleotide, and there's the 2 hydrogen bonds. And this is guanine. See, there's double ring. And then there's 3 hydrogen bonds bonding to cytosine right there. So the pyrimidines are over here, just like it says here, and the purines are here on the right. So that was what Watson and Crick proposed. The tragedy is that it really should have been the Franklin model of DNA. Everybody thought Watson and Crick were gods and that they were geniuses because they figured all this out from practically nothing. As it turns out, they did not use Rosalind Franklin's crummy pictures of the structure of DNA. They used her really high-res photographs of DNA. So let me go to, for you, a picture of what Rosalind Franklin's DNA pictures actually looked like. I go over these in class. So here they are. Rosalind's there on the left. And let's make this big. So there's Rosalind there in her younger days. This is a picture of the DNA that she had released. And you'll notice there's a punch out in the middle, and it's kind of crummy low res. And that's because she didn't want anybody to use the picture to determine the structure of DNA because she was the one taking the risk with the X-rays. Watson and Crick got a hold of high-res photographs where you could see the details and there was no punch-out. So you could actually put a ruler on the dead center of the helix and measure out to these little black bands. Those are the rungs and clusters of atoms that are around the center of the molecule. In this picture here, you're looking down the center of the molecule. You're looking from the end of the molecule down to the other end. You're not looking at the side. So if we go back to that other picture, let's go back to the cursor here so I can get back. If we go back to this outline picture here and go to this picture here, this is looking at the DNA from the side, and so is this. Rosalind's picture here, we're looking at the DNA from the end. So it turned out that Wilkins, a lab, worker with Rosalind, passed information, actual file information, over to Watson and Crick. And instead of using this picture you're looking at on the screen here, they ended up using her high-res pictures. They didn't tell anybody that, of course. They made everybody think that, oh yeah, they used the crummy pictures and they must be gods. And they got the Nobel Prize, and that's what they were looking for. They wanted the Nobel Prize. They were pretty pompous about the whole thing. It's amazing how people will step on other people to get places in science. And at any rate, Rosalind died. She paid the ultimate price for doing her research, and that's what's really annoying. Watson and Crick did nothing, took no chances. Rosalind actually died from probably the X-ray exposure, which induced the breast cancer that took her life in 1958. So enough said about that for right now, or probably there never could be enough said about that. At any rate, now we got to talk about the Hershey-Chase experiment, which is kind of important to nailing the protein theory of DNA as hereditary, nailing the lid down on that. Alfred Hershey and Martha Chase, here's their names here, in 1952, published the results of an experiment where they used viruses. Here's a picture of the viruses here. This is an artistic rendition of, more detail than you're seeing here in the transmission electron micrograph. What these viruses were, though, was just DNA and protein. So this hexagon out here is protein, the blue stuff is protein, the little legs are protein, and the red coil is DNA. What Alfred and Martha did was they labeled the DNA with a radioactive nucleotide, radioactive isotope of phosphorus. And so this in here is radioactive with phosphorus 32, and the protein was radioactive with a different atomic isotope of sulfur 35. So you can tell these two apart pretty easily by their radioactive signatures. What they did then was they allowed radioactive viruses with these two radioisotopes to attack bacteria. Once the viruses attacked bacteria, they wanted to know what ended up in the bacteria, making more viruses. And as it turned out, it was DNA.
So inside the bacteria, they found radioactive DNA. Inside the bacteria, after the viruses had attacked the bacteria, they found radioactive DNA. That pretty much showed that their viruses, which are called bacteriophages, were using DNA as their hereditary substance. And so that by 1952, most biologists were accepting the concept that, okay, it is DNA that's the hereditary chemical, not protein. There's other DNA research you may want to look into. The Matthew Meselson and Franklin Stahl experiment, which nailed down how DNA replication takes place, is kind of an important experiment. And there are others, but we need to move on now to the next topic.